So today in this session we are going to discuss about the bioreactor or fermenter and its conventional design and different types of fermenters. So this is an important topic from Unit 5 of Pharmaceutical Biotechnology of 6th semester B-Pharm. So here it is specified as a general requirement and study of equipment. So the equipment which is used for the fermentation is known as fermenter. So these are some of the questions from this area. This is a very important topic for your examination. And see, you can see as an essay, write in detail about the structure and parts of fermenter, its control with the neat labeled diagram. So diagram is very important here. And second, as an 5-mark question, explain in brief about the structure, parts of fermenter with the help of diagram, and uh, discuss the design features, merits and demerits of continuous fermenters, and answer briefly as different type of sparger and type, control of fermentation parameters. So these are some of the questions. So based on that, this is the learning objective of the session. After the end, by the end of the session, students will be able to understand, describe and write about general requirements for the fermentation, design of an conventional bioreactor or fermenter, and type of bioreactors as well as fermenters. So before starting about the equipments used for the fermentation, let's discuss about the general requirement for the fermentation. So as we mentioned, fermentation is a process of culturing the microorganism in an culture media by using an equipment. So these are the three requirements for an fermentation process also. We need a microorganism which is usually isolated from the soil, lake, ocean, river or plant, animal or air, non-living objects, from any kind of microorganism which have an ability to convert some of the natural components to an related, structurally related compounds. Usually we can use wild strains or improved strains as a microorganism, but by using improved strains by protoplast fusion or mutation or recombinant DNA technology, we can increase the yield of the product. So that is the advantage of improving the strains of the microorganism. And second requirement is culture media. So in order to develop the inoculum and in order to get the desired products, we need to give some kind of nutrients which containing carbon, nitrogen, sulfur, growth factor and antiforming agent containing substances. So usually this will be in natural origin because we want to reduce the cost of the production of pharmaceutical products. So we will go for cheaply available products of natural products, like sometimes we will use starch for production of alcohol, sometimes we will use molasses for the production of alcohol. So these are some of the natural products we can use. And then finally we require an equipment which is known as fermenter or bioreactor, so which will provide for the development of seed culture, fermentation, design of small or large scale production. So these are the general requirement of the fermentation. Anyhow, we will discuss in detail about the three general requirement in detail in further topics. So today's session we will discuss about the equipments which is used for the fermentation process. So as we mentioned, the fermenter or bioreactor is the equipment which is used to perform fermentation or uh, biotransformation and bioprocessing. So we can study only the conventional bioreactor design because it will vary depending on the product which we have to be prepared. So based on that we will vary the design, but we will discuss the conventional design of an bioreactor or fermenter. So I want to inform you one thing. So you can add this kind of conventional design to any question which is related to the fermentation. So if there is any question regarding the fermenter, you must draw this diagram. That is the mandatory thing. If you didn't write anything also, you must draw the diagram of a conventional bioreactor. That is mandatory thing. Without drawing this diagram, you cannot score the mark. Okay, so I repeat, whether it is for 2 marks or 5 marks or 10 marks, you must draw the simple diagram of the fermenter. You don't have to go for this complicated one. You have to draw the simple diagram of the fermenter. Okay, so we can discuss the design of a conventional bioreactor. So before starting to the parts, I want to introduce the fermenter. So this is a basic structure of the fermenter. This is an vessel which is made up of uh, corrosive resistant metals like stainless steel with a shape of doomed shape, which means it is curved in the up and down. Okay, so that is the basic structure, which containing an stainless steel vessel of doomed shape, which means the top and bottom portion is in curved portion. Because why we are keeping this doomed shape? Because it will avoid the accumulation of the any kind of products. It will be very easy to clean the fermenter. That's why we are making this shape into curve. And all this fermenter is jacketed with some kind of jacket through which we can provide some kind of heat or some kind of cold water to control the temperature of the entire process. So that is the cooling jacket or heating jacket. From through this jacket we can provide either steam for heating any kind of conditions, or you can add cold water to reduce the temperature also. And there is an agitator in order to mix the entire product which is there in the fermenter. And there is a part which is known as sparger, which is the facility to provide aeration to the fermentation product or fermenter. So by giving the air through the sparger and by rotating this agitator, the entire product will be mixed uniformly to get an uniform mixture of the microorganism as well as culture media as well as the product. So it will avoid the accumulation of the desired product. It will avoid the accumulation of the toxic products. So I was discussing about the fermenter. So we have already mentioned the role of jacket, role of sparger, and role of agitator. Now there are some connection for acid and alkali antiforming agents, which we will add based on the pH of the entire product which is there in the fermenter. And there is an air exhaust system to remove the air which is there in the fermenter. And there is an medium feed nozzle through which we can add the nutrient media or culture media. And there will be an glass sight glass through which we can monitor the process which is happening there in the fermentation or fermenter. And there is an harvest nozzle through which we can take out or we can remove the final desired product which is there in the fermenter. So these are the basic structure of an bioreactor or fermenter. So this is a three-dimensional structure of the same thing. So this sparger, the agitator will be like this, not an parallel thing, which will be uh, some kind of impeller which will rotate in this fair product or in this mixture of the microorganism, nutrient media as well as the desired product. And this is the sparger, so that we can give the aeration from the upside to uh, sorry, downside to up, so that the aeration will be very effective. So this is the basic structure of the bioreactor or fermenter. So irrespective of the mark of the question, you must draw this simple diagram of the fermenter wherever it is required. Even it is if the question is like structure and parts of the fermenter, or sometime there will be a question production of penicillin by fermentation process. So that you have to, so you have to use this diagram wherever it is required. Now let's discuss in detail about the different parts of the fermenter. So first one is vessel body. So this is the vessel body of the fermenter. So It usually composed of a cylindrical vessel with doomed top and bottom. So I have already mentioned why it is doomed top and bottom, because in order to avoid the accumulation of the products, and usually it is surrounded by the jacket or coil for heating as well as cooling purpose of the entire fermenter. And usually the height to diameter ratio is 2 is to 1, because there will be an double the size of diameter of height will be double the size of diameter. So while seeing this, you can see this is the diameter, so usually the height will be more when compared with the diameter. This is to ensure the efficient mixing and aeration of the fermentation process. That's why it is made in that way, 2 is to 1 ratio. And usually it should be made with the stainless steel, because for its sterility as well as corrosion resistance, and also it must be non-toxic, and it must withstand repeated sterilization with higher pressure steam, and it must withstand higher temperature of 150 degrees Celsius to 180 degrees Celsius, and higher pressure of 377 to 412 kilopascal. So that much should resistance as well as that much should, strength should be there for the material which is used for the manufacturing of the vessel body. Now coming to the second part, that is the agitation system. So this is the agitation system. We have agitators like impeller or agitators, which is usually of a Russian turbine or marine impeller or flat plate, which is mounted on the central shaft. This is the central shaft which is connected to a motor, so that it will be effectively steered in the vessel. Okay, so it will ensure by agitation system we can ensure the uniform mixing of the culture media and also dispersion of the oxygen, and also it will prevent the settling of the cell, and it will maintain is the homogeneity of the entire product which is there inside the uh, fermenter. Now coming to the third part, that is bufflers. So bufflers are nothing but there are some vertical uh, blades uh, which is placed inside the uh, vessels. Okay, vertical strips which is fixed in an inner wall of the vessel, and which is usually used to prevent the vertex formation and to improve the mixing efficiency. Hope you have already aware about what is mean by vertex. So if you are using any kind of stirrer, due to its centrifugal force there will be an formation of hole. Okay, ഒരു വെർട്ടെക്സ് ഫോം ചെയ്യുക എന്നുള്ളത് നിങ്ങൾ കേട്ടിട്ടുണ്ടാവും. So the vertex formation will affect the mixing ability, which will reduce the mixing efficiency. So by preventing the vertex formation, by installing kinds of baffles, we can increase the mixing efficiency of the bioreactors. Now coming to the next one, that is the aeration system. So sometimes the fermentation process has to be carried out in the aerobic conditions by giving aeration like oxygen. So which is usually it will provide sparger at the bottom through which the air such as carbon dioxide, ammonia, or sometimes oxygen can be introduced. So we can use different kinds of spargers to increase the oxygen transfer rate. Okay, oxygen transfer rate, that is the importance of the aeration system. We have to mix oxygen with the entire products. So there are different kinds of spargers. So this is an frequently asked 2 mark question: what is mean by sparger and what are the different types of sparger? So sparger is a device which is fitted at the base of the fermenter to introduce the sterile air or oxygen into the culture media in the form of small bubbles. So this is the sparger facility, which is usually fitted down the or bottom portion of the fermenter to provide sterile air. Okay, so the efficiency of the oxygen transfer usually depend 3 factors. One is bubble size. As the bubble size decreases, as we all know, as the size decreases, there will be an increasing surface area, so that there will be an higher oxygen transfer. And gas distribution inside the vessel also will increases the efficiency. As the gas distribution inside the vessel increases, the mixing efficiency also will increases, and interaction with the agitation system. So if we haven't properly installed impeller inside the fermenter, it can increases the efficiency of oxygen transfer. So these are the three factors which will depends oxygen transfer. So this is the sparger, which is an device fitted at the base of the fermenter to introduce sterile air. Now let's see what are the different types of spargers. So we have porous sparger, orifice spargers, nozzle spargers, and combined sparger, which means sparger with an agitator system. So let's see one by one. First one is porous sparger. So this is usually made up of glass, ceramics, or metal, which have an very small pores in this area. So by giving air through this pipe to this sparger, it will form fine bubbles. So this is an example of sparger. There will be a fine bubble formation through the porous sparger. So it will provide high surface area for gas exchange, but there is an limitation. There is a chance for clogging, and also it is very difficult to sterilize at large scale, and it can be used only for lab scale fermenters of 20 liters 20 liter volume of fermenters. അത്രയും ചെറിയ സ്ഥലത്ത് മാത്രമേ നമുക്ക് porous spargers യൂസ് ചെയ്യാൻ പറ്റുള്ളൂ, because there is a chance for clogging, and also there is a difficulty in sterilization process. So this is the sparger, which is made up of sintered glass, porous sparger, which is made up of sintered glass, ceramics, or metal. So the air passes through the small pores, forming bubbles. Now coming to the orifice sparger, which means this is a simple tube. So there will be several holes or orifice on the simple tube. Sometime it will be in straight line, sometime it will be like in circle form. So this is an example of orifice sparger. Sometimes this is an example of orifice sparger. So there will be many orifice or holes in the sparger. So while providing air through this pipe, the air is released through the hole at the bottom of the fermenter. Usually this will be arranged in the bottom of the fermenter like this. Okay, so that is the orifice sparger. So it will generate large bubbles. That's why that's also they are less efficient in the oxygen transfer, and this is mainly used in the large industrial fermenter, where mechanical agitation will help to break the large bubbles. So even though we are using orifice sparges, we have to use some kind of mechanical agitators, impellers, to mix it properly to reduce the size of the bubble formed through the orifice sparges. Okay, now coming to the nozzle sparger, it is very same as that of the spargers or nozzles we used to use in the gardens. ഗാർഡനിലൊക്കെ നനക്കാൻ വേണ്ടിയിട്ട് യൂസ് ചെയ്യുന്ന സെയിം ടൈപ്പ് ഓഫ് സ്പാർജേഴ്സുകളാണ്. ഇറ്റ്സ് എ സിംഗിൾ open or closed pipe with a downward forcing nozzle. So the sterile air or oxygen is released under the pressure, and it is usually designed to reduce the clogging of the first one, that is that. So that was the porous sparger. So there was a disadvantages of porous sparger. There is a chance for clogging because of its minute holes. But by modifying this into nozzle, which have a bigger size of holes, we can use this as an in large scale production. And also along with this nozzle, we have to use some kind of impellers to reduce the size of the bubble form. Okay, now coming to the last one, that is combined system, which means sparger with the agitator. So this is an example for sparger with agitator. There is a facility to provide aeration in this impeller itself. Okay, in this impeller itself there is a facility to provide, which is usually provided through this central shaft of the impeller. Okay, so by providing this air along with the agitator, it will reduces the size of the bubble formed during this process. So this impeller will break the large bubbles into the small one, so that it will improve the oxygen transfer efficiency. So this is all about the aeration system that is parger, which is used in the conventional fermenter. Now coming to the next part of the conventional fermenter, that is the control system. So we have three type of control system: temperature control system, pH control system, and form control system in the fermenter. So usually temperature control is achieved by the cooling jacket or internal coils. So by using cooling jacket, by providing the cold water to the jacket, we can reduce the temperature of the fermenter. By using internal coil, by using heating coils, we can increase the temperature. So in order to monitor this one, we can use some kind of sensors as well as probes to maintain the optimum growth condition to an fermenter. And coming to the pH control, so there will be an pH probe which will continuously monitor the culture condition. So based on the variation in the pH, we have a facility to add acid or base, which will be added automatically to maintain the desired pH. Now form control, since most of the time we are going to use aerobic fermentation process. So during the use of aerobic fermentation, it is quite natural to have a foam in the products. So in order to reduce the foaming, in order to break the foams, sometimes we may have to use mechanical foam breakers, or we may have to add some kind of anti-foaming agents through sensors, which is uh, facilitated with this control system. So these are the different control system which are there in fermenter. Now coming to ports, there are some ports to add and withdraw some kind of samples. So we have nutrient and inoculum addition port, through which we can add the sterile ports for addition of the feed, inoculum, supplement without any kind of contamination. We don't have to open the large area of the fermenter. We just open the this kind of ports: nutrient, inoculum, and addition port, and we will add the respective thing. And we have some sampling port through which we will periodically withdraw the culture sample, and we will check for its sterility and other conditions in this area. So this is the ports. Now coming to last one, that is exhaust system. So in order to remove the excess gas which is there in the fermenter, we will use exhaust system, but we will use some kind of HEPA filter to prevent the release of microorganism which is added to the fermentation. So these are the different parts of the conventional bioreactors. So there are some other properties must follow for a conventional reactor. So while designing a fermenter, we have to design in such a way that it should be free from kind of services and stagnant area. So that's why I mentioned it should be in doomed shape. ഒരിക്കലും ഒരു സ്റ്റാഗ്നൻ്റ് ഏരിയ, അത്രയും ഷാർപ്പായിട്ടുള്ള കോർണറുകളൊന്നും ഉണ്ടാവാത്ത രീതിയിലായിരിക്കണം ഈ ഒരു ഫെർമെൻറ്റർ നമ്മൾ ഡിസൈൻ ചെയ്യേണ്ടത്, in order to prevent accumulation. Okay, and it should be very easy to clean, and എസ്പെഷ്യലി ദ വെൽഡഡ് ഏരിയ, അങ്ങനെ ജോയിൻ്റ് ചെയ്യാത്ത അല്ലെങ്കിൽ കപ്പിൾ ചെയ്യാത്ത ഏരിയക്ക് പകരം വെൽഡഡ് ആയിട്ടുള്ള, പ്രത്യേകിച്ച് ഇങ്ങനെ എടുത്ത് കാണിക്കുന്ന ഏരിയ ഒന്നും ഉണ്ടാവാത്ത രീതിയിലായിരിക്കണം. ആൻഡ് വി ഹാവ് ടു യൂസ് ദ ട്രാൻസ്പാരൻറ്റ് മെറ്റീരിയൽ wherever it is possible, because we cannot use the transparent material to build the body, but we can use the top of the portion of the fermenter by using transparent material in order to monitor the process which is there inside the fermenter. And there should be a liquid level of a reaction vessel which have a facility for connection for acid, alkali, antifoaming chemicals, etc. So this is all about the conventional fermenter. So you must draw this diagram for irrespective of the mark of the question. എത്ര മാർക്കിന് ചോദിച്ചു കഴിഞ്ഞാലും നിർബന്ധമായിട്ടും ഈ ഒരു ഡയഗ്രാം വരച്ചിട്ടുണ്ടായിരിക്കണം. And depending on the mark and depending on the question, you have to elaborate the different parts of the fermenters in detail. Okay, so this is all about the design of a conventional fermenter. This is a frequently asked question also, so be very thorough with this topic. Now coming to the type of bioreactors or fermenters which is used. So This is the learning objective. After the session, you will able to understand, describe, and write about the different types of fermenters, like stirred tank fermenters, airlift fermenters, fluid bed bioreactor, packed bed reactor, photobioreactors, and membrane bioreactors. So let's see one by one. First one is stirred tank fermenter. As the name indicates, there will be a stirring in a tank of fermenter. That's all. This is the conventional fermenter. There is no change in the conventional fermenter. We have an vessel, we have an impeller. This is the baffle. This is the baffle which is installed inner side of the vessel in order to avoid the vortex formation. So this is the simple conventional fermenter. We will use this stirred tank fermenter mainly for microbial as well as animal cell culture, and it have a function of homogenization, suspension of solid, dispersion of gas liquid dispersion, and it is mainly used for the production of large amount of antibiotic, enzyme, and vaccines, and also it is mainly used in brewing industrial yeast fermentation. So this is all about the stirred tank fermenter. This is nothing but a simple conventional fermenter. Now coming to the second one, that is the air lift fermenter. So there is a difference here. Here we are not going to use any kind of mechanical stirring arrangement for mixing. We are not using any kind of mechanical stirring arrangement. So what we will use? We will use an turbulence by providing air, inlet air from the bottom of the fermenter, which is passes through the draft tube of the central section of the bioreactor. So this is the draft tube. So we will provide the glass flow into this central section of the bioreactor, so that it will flow through this and it will circulate through the entire reactor in upward direction. So that is the difference between the conventional or stir tank fermenter with air lift fermenter. So it is mainly used for both free as well as immobilized cell fermentation process. There are some advantages for this air lift fermenters, because since we are not using any kind of mechanical stirrer, we can eliminate the attrition effect. So what is meant by attrition? It is the normal energy production. There will be a conversion of mechanical energy into heat energy. There will be as an increase in temperature. So if we can, since we are not using any kind of mechanical stirring, we can eliminate the attrition effects which is encountered in the mechanical stirring, and it is most suited for aerobic culture also, and used in mainly to avoid the excess heat produced during the mechanical agitation. So that is the advantage of air lift fermenter. Now coming to the next type of fermenter, that is fluid bed bioreactor or fermenter. So you have already heard about the fluid bed mixer, അല്ലെങ്കിൽ FPD എന്ന് പറയുന്ന സാധനത്തിനെ കുറിച്ച് നിങ്ങൾ കേട്ടിട്ടുണ്ടാവും, fluid bed. Okay, so this is another type of fermenter which is mainly suited to reaction involving fluid suspended particulate catalysts such as immobilized enzyme as well as cell particulate. Comparable to bubble column bioreactor, except the top position is expanded to reduce velocity of the fluid, and it is designed such that the solid are retained in the reactor and the liquid flow out. ആ രീതിയിലാണ് നമ്മളിതിനെ കീപ്പ് ചെയ്തിട്ടുള്ളത്. So liquid flow will be go out, and it is mainly used for carryouting reaction involving fluid suspended biocatalysts such as immobilized enzyme, immobilized cells, microbial floccules. So this is the fluidized bed fermenter. Now coming to the packed bed reactor or fermenter, which consists of bed of solid particle with the biocatalyst or within the matrix of solid packed in the column. So this is the immobilized cell which is packed as an solid particles, and solid is used may be porous or non-porous gel which may be compressible or rigid in the nature, and usually the liquid, the product usually obtained are released into the fluid are removed through the bottom portion. So okay, so that is the packed bed reactor. Nutrient growth is continuously flow over the immobilized catalyst. So this is the immobilized catalyst which is used as an culture media. So the nutrient growth which will be in the liquid nature, which will continuously flow over the immobilized catalyst, and the product usually obtained are released into the fluid and removed. So movement can be upward or downward. Either we can give the inlet here or we can give inlet here. So by giving inlet here, it will require some time to reach the outlet, so that there will be much time for reaction by fermentation reaction, much time കിട്ടും എന്നുള്ളതാണ്. So concentration of the nutrient product can be increased by increasing flow rate of nutrient, uh, by controlling the flow rate of the nutrients. So this is mainly preferred for bioprocessing technology involving product inhibited reactions. So that is the packed bed reactors. And finally we have photobioreactor. So for fermentation, which can be carried out either by exposing sunlight or artificial illumination, can be done in the photobioreactor. Example for the production of uh, para-carotene as well as astaxanthin, ഇങ്ങനെയുള്ള സാധനങ്ങളൊക്കെ പ്രൊഡ്യൂസ് ചെയ്യാൻ വേണ്ടിയിട്ടാണ് ഫോട്ടോ ബയോ റിയാക്ടേഴ്സുകൾ യൂസ് ചെയ്യുന്നത്. Usually these are made up of glass or more transparent plastics. We have array of tubes like this. ഒരു കുറേ അധികം സീരീസ് ഓഫ് ട്യൂബുകളാണ് നമ്മളിവിടെ യൂസ് ചെയ്യുക. So we have array of tube or flat panel constituted light receiving system like the solar system പോലെ തന്നെ ആ ലൈറ്റിനെ റിസീവ് ചെയ്യാനുള്ള ഒരു array of ട്യൂബ്സുകളാണ് നമ്മൾ യൂസ് ചെയ്യുക. And culture can be circulated through the solar receiver by the methods such as using in the centrifugal pump or airlift pump. And it is essential that the cells are continuous circulation without forming any kind of sediment through this kind of tubes. Okay, so that is the bioreactor. So this is mainly performed by exposing the product into the sunlight or artificial illumination. So that is the bioreactor, so photobioreactor. And finally we have the membrane bioreactor, which consists of semi-permeable membrane which is made up of cellulose acetate and other polymeric material, and the primary purpose of this membrane to retain the cell within the bioreactor. So this is an example of membrane bioreactor. So we will have organic phase as well as enzyme activated membrane here, and we will give the aqueous buffer and organic phase reagent, so that we will get the organic phase product as well as aqueous phase product separately from different ends of the bioreactor. So that is the membrane bioreactor. Okay, so that's all about the different types of fermenters. Now coming to the operations of an conventional fermenter. So it is very important for its structural arrangement and also how it operate. ഇത് രണ്ടും മനസ്സിലാക്കുക എന്നുള്ളത് വളരെ ഇമ്പോർട്ടൻ്റ് ആണ്. So what are the different operation which has to be carried out? So this is also a frequently asked question, like control. Its control, you can see the detailed structure and parts of fermenter and its control. And also there are some questions like control of fermentation parameters. So let's see what are the different control parameters here. So this is the learning objective. After the session you will be able to understand, describe about the different operations of conventional fermenters like sterilization, inoculation and sampling, aeration and control systems, and cleaning. So these are the different operations of a conventional fermenter. So let's see one by one. First one is sterilization. So in order to produce any kind of things in foreign pharmaceutical purposes, it is very mandatory to keep it sterile. So it has to be performed in aseptic condition. So this is the basic requirement for a successful fermentation. And usually sterilization is very essential to ensure the sterility of the culture media, ensure the sterility of incoming and outgoing airs, ensure to sterility of bioreactor, and also for the prevention of contamination during the fermentation process. So first let's see how we can sterilize the culture media. We have already studied the culture media are usually sterilized by the autoclave method, and sometimes culture media containing some vegetative cells or some kind of what we will thermolabile substances, we may have to go for some kind of other methods. So usually we used to sterilize culture media by applying heat, to lesser extent by using like some kind of physical method, chemical method, or radiation method. And most economical and most cheap method is always it will be heat. Now, what are the different physical method for sterilization of the culture media? You can go for filtration, centrifugation, or adsorption, and it is mainly used for heat-labile constituents such as vitamin, blood component, and antibiotic. Otherwise, we will go for the autoclave. Only if the culture media containing any kind of heat-labile constituents such as vitamin, blood components, and antibiotic, we will go for the filtration, centrifugation, or adsorption. But there are some limitation for the filtration. We cannot give high pressure of filtration because sometimes we may have to increase the speed of the process. So in order to increase the speed, we may have to give the pressure. So it is not possible in case of filtration process, and some of the media component may be lost during the media during filtration. So that also not a good thing for an sterilization of the culture media. And chemical method usually by using some kind of chemicals such as disinfectant, but these are not preferred in case of fermentation process. And we will use some type of radiation procedure like by using UV rays or gamma rays. X-rays are not commonly used for the media sterilization, but this will be costly when compared with the conventional heating method. So we go for the heat sterilization method. This is the most widely used method, and important factor which will affect the sterilization are quality and quantity of the contamination, composition of the media, pH, and size of the suspended particle. So usually by giving an temperature of 60 degrees Celsius for 5 to 10 minutes, we can kill vegetative cells. But the spore, if there is a spore in the spore in the fermenter, we may have to heat to 80 degrees Celsius and above for 50 to 20 minutes. So in order to ensure the successness of the sterility testing or sterilization process, we can use Bacillus thermophilus, which is a moisture resistant strains of a microorganism, so which is mainly used as a biological indicator for assessing the efficiency of the sterilization process. Okay. Now, it can be done by batch or continuous method. So, by batch sterilization process, what we will do, the culture media are subjected to sterilization at 121 degrees Celsius in batch volume, and usually this can be done by injecting steam into the medium by direct method, or injecting steam into the interior coil by indirect method. So that we can perform the batch sterilization method either by direct method by injecting the steam directly into the culture media, or by indirectly injecting the steam into the interior coil, interior jacket of the fermenter. So these are the batch sterilization process, but there are some disadvantages. It may damage culture media, and high energy consumption is required. So these are the disadvantages of batch sterilization. Now coming to the continuous sterilization process, here it is carried out at 140 degrees Celsius for a short period of 20 to 120 seconds, based on the principle that time required for killing microorganism much shorter at higher temperature. And this also can be done by injecting steam into bioreactor or by means of heat exchanger. So there are three type of heat exchanger are used for this kind of continuous sterilization process. So first heat exchanger will raise its temperature to 90 to 120 degrees Celsius in 20 to 30 seconds. In second exchanger, it will further increase its temperature to 140 degrees Celsius and maintain for 30 to 120 seconds. And third exchanger will bring down the temperature by cooling to in next 20 to 30 seconds. So this is the method of continuous sterilization by using heat exchangers. So that's all about the sterilization of the culture media. Now we have to ensure the sterility of the air which is used for the fermentation process. So it can be done by filtration, heat, UV radiation, or gas scrubbing. So usually air sterilization by heat is done by passing through the electrically heated element. Uh, but since this is much expensive, ബിക്കോസ് എയറിനെ എത്ര പെട്ടെന്ന് സ്റ്റെറിലൈസ് ചെയ്യാന്ന് പറഞ്ഞാൽ എളുപ്പമല്ല. ഈ എയർ നമുക്ക് കാണാൻ പറ്റാത്തതാണ്, മോണിറ്റർ ചെയ്യാൻ പറ്റാത്തതാണ്. സോ യൂഷ്വലി ഫോർ സ്റ്റെറിലൈസേഷൻ ഓഫ് എയർ വി കോമൺലി യൂസ്ഡ് filtration sterilization for air. So by using depth filters or membrane cartridge filters. So in case of depth filter, air is passed through the glass pool containing the depth filters. So there will be an physical efforts such as inertia, blocking, gravity, electrostatic attraction, and diffusion. So these are the different process of depth filters. And membrane cartridge filters, usually by using a membrane filter made up of cellulose esters, nylon, or polysulfone. So this is the method for sterilization of the air. Now sterilization of the bioreactor. So we have already sterilized the culture media, we have sterilized the air. Now we have to sterilize the equipment which is used for the fermentation process, and this can be done by in situ sterilization or by continuous heat sterilization, which is just as that we have mentioned for the culture media. We have to inject the pressurized steam into the fermenter by injecting the steam at 120 degrees Celsius for 20 minutes, and or by increasing the temperature of 150 degrees Celsius for 120 seconds. That is the two method of sterilization of bioreactors. So that is the first step, sterilization. Before starting the fermentation, we have to sterilize a fermenter, we have to sterilize the culture media, we have to sterilize the air. Now coming to the second step of the fermentation process, the inoculation and sampling. So basically we will take the culture media which is sterilized, and then we will inoculate the culture media with a microorganism which have an ability to convert the culture media used to a structurally similar desirable product. ഇപ്പോൾ നമ്മൾ എടുക്കുന്ന നാച്ചുറൽ ആയിട്ടുള്ള എന്താണോ മെറ്റീരിയൽ എടുത്തിട്ടുള്ളത്, ആ മെറ്റീരിയൽ സ്ട്രക്ചറലി സിമിലർ ആയിട്ടുള്ള ഡിസൈറബിൾ ആയിട്ടുള്ള ഒരു പ്രൊഡക്റ്റ് ഉണ്ടാക്കാൻ വേണ്ട ഒരു മൈക്രോ ഓർഗാനിസത്തിനെയാണ് നമ്മൾ ഇനോക്കുലേറ്റ് ചെയ്യാൻ പോകുന്നത്. ഓക്കെ, നോ എത്ര ml ആണ് നമ്മൾ ഇനോക്കുലേറ്റ് ചെയ്യുക എന്നുള്ളതാണ്. സോ we have to take almost 1 to 10% of the total volume of the media. So suppose you are going to take 1000 ml of the culture media, we will take 100 ml of the inoculum. That is the 1 to 10% means. If you are taking 1000 ml, you will take 100 ml of the culture media. If you are going to take 1 lakh liter of the culture media, we will take almost 1 lakh. Okay, so the 1 lakh volume. So that is the 10% of the volume of the media. So a high yielding product strain of a microorganism is usually taken. So sometimes there will be same microorganism with a different strains, and many strains will give different types of yield, percentage yield. So we have to take an strain which have an high yielding production, high yielding ability. Okay, then after inoculating, we will give the sufficient environment for the growth of the microorganism, and during this period at regular intervals we will draw some sample in order to check the contamination and also to measure the product form. So suppose we have to carry out fermentation for 10 days. So each day we have to take the sample from the fermenter, and we have to check for whether there is an contamination by any kind of other microorganism, and how much of the product is formed in the culture media. This we have to measure by sampling at regular intervals. So that is the second operation which will happen in the fermentation process, that is inoculation and sampling. Now coming to the aeration. So this is another important factors which is required for the fermentation. So we have to give aeration by supplying oxygen, and we have to remove the carbon dioxide which is formed during the bioreaction. So by using sparger, we will provide the oxygen, and by using the exhaust facility, we will remove the unwanted gases. So usually, if you are taking an fermenter, 20 percentage of the total fermenter volume will be remain vacant. Usually that is known as head space, and this living of this space is for the accumulation or for keeping the unwanted gases. അതിനു വേണ്ടിയിട്ടാണ് നമ്മൾ ഇപ്പോൾ 1000 ലിറ്ററിൻ്റെ ഒരു ഫെർമെൻ്ററാണ് നമ്മൾ യൂസ് ചെയ്യുന്നതെന്നുണ്ടെങ്കിൽ അതിൽ കൾച്ചർ മീഡിയ നമ്മൾ 80% എടുക്കുള്ളൂ. 1000 ലിറ്റർ ആണ്, 800 ml നമ്മൾ കൾച്ചർ മീഡിയ എടുക്കുള്ളൂ. ആ 800 ml 10% നമ്മൾ ഇനോക്കുലം ആഡ് ചെയ്യുള്ളൂ. ഓക്കെ, സോ പ്ലീസ് നോട്ട് ദാറ്റ് വൺ. വി വിൽ നോട്ട് ഫിൽ ദ കൾച്ചർ മീഡിയ, we will not fill the fermenter with the culture media. The entire volume will not be filled. We will keep it the 20% of volume as vacant space, okay, for aeration. Now there are different type of aeration procedure. We can give airlift system of aeration, which means we have to give aeration at the bottom. So by giving this airlift formation, aeration capacity will be increased, and also it is there are some factor, airflow rate as well as internal pressure are the two factor which is directly proportional to the aeration. As the aeration flow rate increases as well as the internal pressure increases, aeration capacity will increases. And second type of aeration is stirred system of aeration, which means we have to give air in the bottom, and we have to use some kind of stirrer or impeller for mixing this or stir this oxygen which is provided to the bottom of the media. So there are some kind of porous spargers as well as orifice sparger which can be used for aeration. Now coming to the fourth operation, that is the control system. So while performing the fermentation process, we have to control many factors to get the desirable product for at an higher yield. So usually these are the some of the factor we have to control: pH, temperature, dissolved oxygen, adequate mixing, nutrient concentration, and form formation. So basically the pH we have to keep between 5.5 to 8.5, which means we cannot perform fermentation in extremely acidic condition or extremely basic condition. So we have to keep the pH in between the extreme acidic as well as extreme basic condition. And what is the reason for changing the pH? Because as the process occur from the lag phase to log phase to stationary phase to decline phase, there will be some metabolic release of metabolic rate or release of metabolic product which will changes the pH inside the fermenter. So we have to monitor the change in pH, and we have to maintain the optimum level by adding the respective thing. If it is too acidic, we have to add basic alkali, and if it is too alkaline, we have to add acid to neutralize to this range 5.5 to 8.5. Now coming to the second factor, that is the temperature. So again, temperature control is absolute essential for good fermentation process, since the lower temperature will causes the reduced product formation, and higher temperature will causes the adverse effect on the growth of microorganisms. So as the temperature increases, microorganism got killed. Okay, so what will happen? There will not be any further fermentation process. But what will happen if there is a lower temperature? It will reduces the product formation. Okay, so we have to keep the temperature in between these two levels. It should not be too low and it should not be too high. So by using cooling jackets or by using heating coil, we have to adjust the temperature, and we will monitor the temperature by using sensors in the temperature. Now coming to the next factor, that is the dissolved oxygen. So continuous oxygen supply is very essential for the working of the oxygen, working of the entire process, and it can be done by using different aeration system like spargers. Now adequate mixing, another important factor which has to be considered. So by using stirrers, homogenizers, or anything, we have to keep stirring in order to avoid the accumulation of the toxic product, in order to avoid accumulation of the desired product, in order to avoid accumulation of the fresh nutrient added to the fermenters. So everything should be mixed properly to get uniform solution of the microorganism, culture media, and product. Okay, so all these things should be adequately mixed in order to get an proper uniform structure. Now nutrient concentration again, as the fermentation progresses, sometimes the microorganism will uses the nutrient and it will start producing the metabolic end products. So metabolic end products will cause adverse effect into the organism, and it will further decreases the production of the product or desired products. So by adding limiting concentration or by adding the required nutrients at specific time, we can control the product formation. So in usually by adding higher concentration of the nutrient media, it has an inhibitory effect. We do not think by giving more nutrient into the fermenter we can get more yield of the desired product. It's not like that. Higher concentration is usually associated with the inhibitory action. So we have to give the nutrient in limited concentration in order to get on optimum product formation. So we have to maintain the optimum nutrient concentration. So we will monitor the nutrient concentration and product formed through the regular interval by sampling. And finally the formation, since we are using proteinaceous materials or media, and also we are performing the fermentation in the aerobic condition, sometimes there will be a formation of foams. So we can reduce the foam formation by using anti-foaming agents like mineral oil based silicone or vegetable oils, or by using mechanical foam bakers in order to reduce the foam formation. And finally the last step is cleaning. So once the fermentation process is over for specific period of 5 to 10 days, all the content are removed. So this removal of content from the fermentation is known as harvester. നമ്മൾ എന്തിനാണോ ഈ ഒരു പ്രോസസ്സ് ചെയ്തത്, ആ പ്രോസസ്സിൻ്റെ ഫൈനലി കിട്ടുന്ന പ്രൊഡക്റ്റിനെ നമ്മൾ എടുക്കുക എന്ന് പറയുന്ന പ്രോസസ്സിനെയാണ് നമ്മൾ ഹാർവെസ്റ്റിംഗ് എന്ന് പറയുക. സോ ദ റിമൂവൽ ഓഫ് ദി എൻറ്റയർ കണ്ടന്റ് ഈസ് നോൺ ആസ് harvesting. Now after removing the product from the fermenter, we have to make ready the fermenter for the next batch of fermentation process. So that is known as turn around. ആ ഒരു പ്രോസസ്സിനെയാണ് നമ്മൾ ടേൺ അറൗണ്ട് എന്ന് പറയാം. ഒരു പ്രോസസ്സ് കഴിഞ്ഞ് ക്ലീൻ ചെയ്ത് അടുത്ത പ്രോസസ്സ് തുടങ്ങുന്ന ആ ഒരു പ്രോസസ്സിനെയാണ് ടേൺ അറൗണ്ട് എന്ന് പറയുന്നത്. ആൻഡ് ദ ടൈം റിക്വയർഡ് ഫോർ ടേൺ അറൗണ്ട് ഈസ് നോൺ ആസ് ഡൗൺ ടൈം. ഒരു പ്രോസസ്സ് കഴിഞ്ഞ് അടുത്ത പ്രോസസ്സ് തുടങ്ങാൻ വേണ്ട സമയത്തിനെയാണ് നമ്മൾ ഡൗൺ ടൈം എന്ന് പറയാം. സോ വി ഹാവ് ടു plan and fermentation process with a minimum downtime. It should be very easy to remove, it should be very easy to clean, and it should be very easy to make ready for the next batch of fermentation. So in order to clean this kind of fermenter, we may have to use some kind of high pressure water jet from nozzle fitted inside the reaction vessel, because we cannot enter into the bioreactor. So it should be very automatically cleaned by using high pressure water jet from the nozzle, which is fitted inside the reaction vessel itself. So that is the cleaning method. So that's all about the fermenter, types of fermenters, and different operations of fermenters. So this is a very important topic. So there are many questions from this area. Hope you have already aware. So I have already shown you different questions from this area, like detailed structure and parts of the fermenter and its control with a neat labeled diagram, and explain in brief about the structure and parts of fermenter with the help of diagram. Discuss the design, structure, merits and demerits of continuous fermenters, and different types of spargers, charger and types, and control of fermentation parameters. So this is all about the conventional design of an bioreactor, then different types of an bioreactors and operations of an bioreactors. So hope it is clear. Thank you for your patient listening.